Assalamu alaikum. I am Ahmed Yassin and I want to talk about development of the lung buds and trachea, esophagus and some abnormalities during the development. Formation of lung buds. When the embryo is approximately 4 weeks old, the respiratory diverticulum or lung bud appears as an outgrowth from the ventral wall of the foregut. Here, the yellow pipe is the foregut, and here the blue one is the respiratory diverticulum or lung bud. Now, when the diverticulum expands caudally, two longitudinal ridges, tracheoesophageal ridges, separate it from the foregut. Subsequently, when these ridges fuse to form the tracheoesophageal septum, the foregut divides into a dorsal portion here, which is the esophagus, and ventral portion, which is the trachea and lung buds. Respiratory primordium maintains its communication with the pharynx through laryngeal orifice. At first, the esophagus is short, but with descent of the heart and lungs, it lengthens rapidly. The muscular coat, which is formed by surrounding splanchnic mesenchyme, is striated in its upper 2/3 and innervated by the vagus. The muscle coat is smooth in the lower third and is innervated by splanchnic plexus. Now let's talk about esophageal abnormalities. Esophageal atresia and or tracheoesophageal fistula results either from spontaneous posterior deviation of the tracheoesophageal septum or from some mechanical factor pushing the dorsal wall of the foregut anteriorly. In its most common form, the proximal part of esophagus ends as a blind sac, like here, and distal part is connected to the trachea by a narrow canal just above the bifurcation. Other types of defects in this region occur much less frequently. Now let's see another form of esophageal atresia. Here is a blind sac in the proximal part and a blind sac in the distal part, and this form occurs in approximately 4% of newborns. There are a lot of other forms, like communication of esophagus with trachea with common canal, like this. Here, proximal and distal parts are connected to trachea by communication in a common canal, or the proximal part is connected to the trachea and distal part is not connected with the trachea, and this also occurs in approximately 4% of population. Other form, that the proximal part is connected to trachea in a distant canal and distal part is connected with distinct canal. Atresia of the esophagus prevents normal passage of amniotic fluid into the intestinal tract, resulting in accumulation of excess fluid in the amniotic sac, which is called polyhydramnios. In addition to atresias, the lumen of the esophagus may narrow, producing esophageal stenosis, usually in the lower third. Stenosis may be caused by incomplete recanalization, vascular abnormalities, or accidents that compromise blood flow. Occasionally, the esophagus fails to lengthen sufficiently, and the stomach is pulled up into the esophageal hiatus through the diaphragm. The result is a congenital hiatal hernia. Now I want to talk about maturation of the lungs. Up to the 7th prenatal month, the bronchioles divide continuously into more and smaller canals, which is called canalicular phase, and the vascular supply increases steadily. Now we see here, these are blood capillaries, and this is respiratory bronchioles, and these cells are cuboidal epithelial cells, and this is the terminal bronchioles. Respiration becomes possible when some of the cells of the cuboidal respiratory bronchioles change into thin, flat cells, like this. Flat and thin cells. Flat and thin cells. Flat and thin cells. These cells are intimately associated with the numerous blood and lymph capillaries, and the surrounding spaces are now known as terminal sacs. Now these are called terminal sacs, and as we said, numerous blood capillaries, like this. During the 7th month, sufficient numbers of capillaries are present to guarantee adequate gas exchange, and the premature infant is able to survive. During the last 2 months of prenatal life and for several years thereafter, the number of terminal sacs increases steadily. In addition, cells lining the sacs, known as type 1 alveolar epithelial cells, these cells now are known as type 1 alveolar epithelial cells, become thinner so that surrounding capillaries protrude into the alveolar sacs. They will make invagination inside them. This is the alveolar period from 8 months to childhood. We see here mature alveoli which have well-developed epithelial endothelial capillary contacts. Mature alveoli are not present before birth, in addition to endothelial cells and the flat alveolar epithelial cells. Another cell type develops at the end of the 6th month. These cells, type 2 alveolar epithelial cells, produce surfactant, a phospholipid-rich fluid capable of lowering surface tension at the air-alveolar interface. Before birth, the lungs are full of fluid that contains a high chloride concentration, little protein, some mucus from the bronchial glands, and surfactant from alveolar epithelial cells type 2. The amount of surfactant in the fluid increases, particularly during the last 2 weeks before birth. As concentrations of surfactant increase during the 34th week of gestation, some of this phospholipid enters the amniotic fluid and acts on macrophages in the amniotic cavity. Fetal breathing movements begin before birth and cause aspiration of amniotic fluid. These movements are important for stimulating lung development and conditioning respiratory muscles. When respiration begins at birth, most of the lung fluid is rapidly resorbed by blood and lymph capillaries, by these capillaries and lymph capillaries, and a small amount is probably expelled via the trachea and the bronchi during delivery. When the fluid is resorbed from alveolar sacs to the capillaries, surfactant remains deposited as a thin phospholipid coat on alveolar cell membranes on these cells. With air entering alveoli during the first breath, The surfactant coat prevents development of an air-water interface with high surface tension. Without the fatty surfactant layer, the alveoli would collapse during expiration, which is called atelectasis. Respiratory movements after birth during bring air into the lungs, which is expand and fill the pleural cavity. Although the alveoli increase somewhat in size, the growth of the lungs after birth is due primarily to an increase in the number of respiratory bronchioles and alveoli. It's estimated that only 1/6 of adult number of alveoli are present at birth. The remaining alveoli are formed during the first 10 years of postnatal life through the continuous formation of a new primitive alveoli. I hope that you understood and was interested by what I explained. Thank you for listening and best wishes. Thank you.